presentation today.
You want to see everybody at the box.
And
Great.
I want to start off by thanking everybody for coming. And we have a great talk today on ticks. We are lucky to be joined by Dr. Kirby Stafford. Dr. Stafford, of course, is the Chief Science State Entomologist since 2013. Also means that he's the State Plant Regulatory Officer. Dr. Stafford is a medical veterinarian entomologist who researches focus on the ecology and the control of the black-legged tick. So we couldn't think of anybody better to help us talk about our ticks. He's been studying on this since 1987. And with that, I think we'll start right in. Well, welcome.
Hey, thank you, Brett, and welcome everyone. Good afternoon. Under normal circumstances, I'd be giving this presentation at your actual meeting this evening, with perhaps just a little more time to cover a little more material. But let's go ahead and get started. I'll probably be going pretty quickly. And now it doesn't want to move. There we go.
There we go. Yeah.
Okay, so I'll start off just pointing out that there are a lot of tick-associated diseases of humans in the U.S. There's 15 to 20 of them. I'm not going to have time, obviously, to go over these in any detail, but the ones that are in blue are the ones that are associated with our black-legged tick or deer tick. Of course, Lyme disease is the major one. And if you look at all of those diseases in total, you can see that there's been a steady increase in the total number of tick-borne disease cases here in the United States. This chart shows from 2004 to 2017, and those numbers just continue to go up. And these are the actual documented or reported ones. Lyme disease, of course, comprises the bulk of those cases. In 2017, it was 68% of them. With over 42,000 cases, and other tick-borne diseases, 27%. And if you'll note that the mosquito or flea-borne diseases was only a little itty-bitty 5%. But only about 10% of the diagnosed cases actually end up being reported, so the actual number true number of Lyme disease cases probably runs somewhere between 330 and 430,000 cases every year. It's a lot of Lyme disease. Now here in Connecticut, we have at least 16 species of ticks, 3 species that commonly bite humans, and 4 species that can transmit disease pathogens. And the 3 common ones, of course, is our black-legged tick, Ixodes scapularis, or the deer tick, as many people know it, the American dog tick, Dermacentor variabilis, and increasingly of importance, which I'll go over a little bit later, the Lone Star tick, Amblyomma americanum. We get a few people bitten by the woodchuck tick, Ixodes cookii. And of course, we do see occasional exotic tick species brought in from people that are doing foreign travel, you know, maybe going on safari in Kenya or something like that. And they actually come back to the U.S. and find they have a tick on them. And then we have a new invasive Asian.
I just heard the meeting for Connecticut Port. And look, I could be in there if I clicked, but I don't want to be in there. And these are all the people.
Is this present time?
Yeah, right this minute. It started at 4:30.
Are those people?
They're right. They're on Zoom.
Looks familiar.
That's Brett from Connecticut Port.
Oh,
Brett Isaacson.
All right. So as a reminder, let me just point out that this tick is a 3-host tick. There are 3 active stages, the larva, the nymph, and the adult. And as you'll see in this chart, the larval ticks hatch from the eggs. They feed predominantly on rodents and birds, but the black-legged tick will feed, larvae will feed on us as well. They are not infected, okay? So the Lyme disease pathogen and most of the others are not transmitted by the female.
It's so cool that you can use someone like this speak. You know,
and
this is through Zoom.
Someone's talking. Thank you. And then once they feed, they drop off, becomes a nymphal tick. The nymphal tick, again, feeds predominantly on birds and rodents, but it'll feed on anything, including us. Then once it feeds, it drops off, becomes an adult tick, which only feeds on medium to large animal hosts. And then in each female tick, when she drops off, primarily deer, will lay about 2,000 eggs. So it's what we call a 3-host tick life cycle. And you can see from the top to bottom there on the right, the larva, the nymph, the female, as well as an engorged female with her eggs. So the critical thing when you're out thinking about your risk, Lyme disease risk, the question is, where can you pick them up? When will you pick them up? And what their prevalence of infection rate is in those ticks. So this is some results from a survey that was done by the Stanford Health Department many years ago. And the questions they asked for ticks that were being submitted to the experiment station for testing is, what were you doing or where were you when you picked up the tick? And 74% picked up the tick right at home. 21% picked it up away from home. What do you think you were doing when you picked up the tick? Well, you'll notice 47% were associated with play outdoors, and indeed the highest rate of Lyme disease actually is in children. But you'll see the other outdoor activities, high risk, gardening, 12% of the submitted cases, yard work, 18%. So, which makes sense. I mean, that's where you're going to be exposed to them. The other aspect is a seasonal activity of this tick. So it's a 2-year life cycle. And so the adult ticks female ticks will lay their eggs in May. The larval ticks will hatch from those eggs in the late summer, around mid-July, with August being the peak month for larval tick activity. Once those larvae feed on their hosts, like I said, primarily mice and birds, they drop off, molt the nymphs. They will go through the winter as nymphs, inactive, and then appear the following year, generally in May, but we're starting to see a few already. And so late May, June, and July is the peak season for nymphal tick activity here. And then once those nymphs feed, they drop off. They will appear in the fall as adults. The adults do not hibernate. They will be active even through the winter months if the temperatures are mild enough. And then we see kind of a secondary peak in the spring as activity picks up again. So the spring adults that you're seeing now were adults last fall as well. So if you overlay that month seasonal activity with the month of onset for Lyme disease, you will notice the correspondence between the curve for nymphal tick activity and the majority of Lyme disease cases. That small summer nymphal stage is responsible for the bulk of Lyme disease cases. It's obviously when you're all outdoors, and the nymph is very small and a lot easier to miss when you're doing a tick check. So what is your risk from any one particular tick? Okay, so we do the tick testing service for residents of Connecticut. These are the results of ticks received for testing in 2019. You'll see that we received over 3,700 black-legged ticks, almost 750 American dog ticks, a handful of Lone Star ticks, 145 of those, which comprise 3.1% of the total, and then a few other species, like 3 of those Ixodes cookii woodchuck ticks, and so on. So we had over 4,000 ticks that were submitted to the tick testing laboratory, and the majority of those submissions came from New Haven and Fairfield County. This is the results of the tick testings from 2015 to 2019. And the point I want to make out, if you'll look at the second line, a number uninfected out of the thousands tested, you will see that it is running roughly around half. I think it actually comes to about 50 to 53% of the ticks were uninfected. So that's an important point. Any tick you pick up, you've got a 50-50 chance at the beginning whether that tick is infected or not. And you'll see that roughly between 30 to almost 40% of those ticks, and this is both nymphs and adults, were infected with Borrelia burgdorferi, the causal agent of Lyme disease. And then a smaller proportion were infected with the organisms that causes babesiosis and anaplasmosis. So here we're ranging below 10% or down to 5%. And then there's a smaller proportion of ticks that are co-infected. So it's actually possible for you to get Lyme disease and human babesiosis from the same tick bite. But you saw how the samples that we get are heavily biased toward Fairfield and New Haven counties. So in 2019, with some funding from the CDC channel through the Department of Public Health, we initiated an active tick surveillance program with 40 locations through all 8 counties of the state. And that means instead of waiting for people to send ticks in for testing, we're actually going out and collecting them. We collected over 2,000 ticks in 2019, black-legged ticks, over 400 American dog ticks, 3 Lone Star ticks, and 2 Asian Longhorn ticks. And they were all tested here. Here you will see the results of that testing. And you'll see, even though in that earlier graph you saw broadly how many ticks were infected with the Lyme disease agents, Borrelia burgdorferi, you will see here there's quite a difference between the infection rate in the adult ticks. See that 45% of the females were infected versus the nymphs, which was only 15% last year. And that's because the 2-year life cycle, when the ticks feed, they have 2 chances to become infected. So infection rates tend to be higher in the adults.
Dr. Stafford, could
Yes.
I get you to just click on the participants and let the other people that are waiting in?
More.
Yeah.
Okay. There we go.
Awesome. Thank you.
You're welcome. Okay. So one of the things I want to bring up, something to be aware of, is the Lone Star tick. This tick is responsible for over 90% of the tick bites in the southeastern United States. And it is a vector for a different suite of diseases, mainly ehrlichiosis, but some viruses, tularemia, heartland virus, some spotted fever, group rickettsia, tularemia. But what really gets people kind of upset or concerned tends to be the red meat allergy. There are indications that this tick can actually introduce a allergic reaction to red meat due to a sugar that is found in mammals, but not in us, not in primates. And so it's possible to develop an allergy to that particular sugar. And if you do develop that allergy, you may not be eating any steaks or hamburgers again. It's relatively new and not a lot is really known about it. And we don't know whether that allergy is permanent or not. Here in Connecticut, we've been seeing a steady increase in the small but steady and significant increase in the number of Lone Star ticks that are being submitted to the tick testing lab, and the majority of those have come from Fairfield County. And it was back in 2017 that we had a report of a dead and dying deer on Manresa Island in South Norwalk, Connecticut. The picture, the upper picture on the right, was sent to me by a environmental conservation officer. I met him the next day. This deer, unfortunate animal, had been completely covered with Lone Star ticks. We had a very isolated but extremely high population of Lone Star ticks here. So we're starting to see an increase in the number of Lone Star ticks here in the Northeast and along the Atlantic seaboard. They actually first started moving into Long Island in 1990. They're extremely abundant on parts of Long Island. It's a very aggressive tick. And as we think as the current environmental and climate conditions are becoming more moderate, it's allowing this tick to expand northward. And we've actually done studies where we have found that this tick can survive the winter here in Connecticut. And a very small proportion actually survived up in Maine as well. It's a very aggressive tick species. Another tick that we're keeping our eye on is the Asian longhorn tick, Haemaphysalis longicornis. This tick was first discovered on a sheep in a farm in Hunterdon County, New Jersey, back in November of 2017. This East Asian tick is a serious pest of livestock, including cattle, horses, sheep, and goats. It will attack humans to some extent, but also pets. It likes dogs and wildlife, likes deer. It is a known vector for a number of human and animal pathogens in its native range, which is the Korea, northeastern China. It got introduced into Australia and New Zealand around the turn of the early 1900s and late 1800s, where it's, again, a major livestock pest. So far, this tick has been detected in 12 U.S. states, ranging from Tennessee up here to Connecticut. And I'm sure we're going to find more of it. In fact, there were some cattle that were reported killed due to the tick load by this tick. This tick is very abundant on Staten Island and parts of Westchester County. We've only found a few in Connecticut so far. But it's definitely something we're keeping an eye on. The unique thing about this tick is there's no male ticks. It reproduces parthenogenetically. So that means that a female tick does not need to mate, and it can just, once it feeds, it can lay its eggs and build up a population very, very, very quickly. So it remains to be seen what the full impact of this tick will be. But it's certainly going to be of high concern, particularly for the livestock industry. Okay, so that's a quick summation of the ticks. And so what I want to do now is just briefly go into what do we do about them. So I've been doing a lot of research on what I'll loosely call integrated tick management. And some of the things that we've looked at are personal protection measures, landscape modifications to make the habitat less suitable for the tick. You have chemical control with synthetic insecticides. We looked at a variety of botanical or so-called natural compounds, biological control, host reduction or exclusion, for example, fencing out deer, which is quite effective actually. Host-targeted acaricides, which means treating the deer or mice. And we're starting to look at host-targeted vaccine. There's actually a vaccine, an oral bait vaccine that's being developed that we've done some testing with for white-footed mice, which is the reservoir host for Lyme disease. It's where the ticks get it. The ticks give it to the mice give it to a new generation of ticks, which then infect the new generation of mice. Deer is the main reproductive host for the tick. Remember I said the adult females are feeding on the deer. So deer basically determine how many ticks you have. They're not involved with the disease cycle. And the mice then and chipmunks determine how many are actually infected. So your first line of defense is your personal protection measures. Wear clothing, pants tucked in the socks. I know it looks dorky, but I do it. You can use a skin-based repellent like DEET. You want to use around 25 to 30%, or picaridin 20%, or oil of lemon eucalyptus 30%. The lower concentrations available that might be a little effective for mosquitoes won't be quite as effective for ticks. What's really effective is the permethrin-based clothing tick repellent. It's 0.5% permethrin. It's extremely effective. It's what I use, and I haven't gotten Lyme disease yet. You can also buy from several companies pre-treated permethrin clothing. You know, the pants, the sock shirts, and so forth, and those will last at least 50 washings. And then when you come back, bathe and definitely do your tick checks. So when you're out taking your hike, and I know right there in this time, there's a lot of people enjoying the outdoors right now after being cooped up with the coronavirus stay-home precautions. And then when you try to get out, in fact, some parks were closed because there were too many people getting out in the same location. But just think about where you're walking. You want to use tick bite prevention as well as keeping that social distancing. And this is just an example of what I would consider a more tick-safe trail. They're wide, well, kind of paved, that you can kind of keep away from the edge of the vegetation where you might pick up a tick, versus the cautionary trails where your high risk would be quite high for encountering a tick. And obviously the two people you see here are not being cautious in terms of tick bite prevention measures. Once you come back and do a tick check, you want to remove the tick. I prefer fine tip forceps to grasp the tick as close to the skin as possible and gently and firmly pull it out. The nymphal tick is quite small. This is a picture of an actual female tick attached, and then a drawing of an engorged nymphal tick with a straight pin by comparison. So even fully engorged, a nymph is no bigger than the head of a pin. And you can also see the ticks in my hand. So the tick checks are a challenge, particularly in the summer when you're looking for those very tiny nymphs. It takes over 24 hours for an infected tick to transmit the Lyme disease spirochetes to you. So even if the tick is infected and you get it out within the first 24 to 36 hours, you won't get Lyme disease from that particular tick. So another approach is looking at landscape or vegetative management. The ticks do require pretty high humidity and cover for survival. And I just kind of a little aside, New England has 240,000 miles of stone walls, and they're all mouse hotels. So there's a lot of great habitat out there, which is one of the reasons we have a big tick problem. The one thing in this talk I'll kind of mention in terms of vegetation is Japanese barberry. The abundance of adult black-legged ticks that are infected with Brilobigdoferi can be extremely high in areas of dense Japanese barberry, which is, I think most of you know, invasive. Similarly, a greater number of Lone Star ticks were infected with Ehrlichia, were present in dense stands of honeysuckle. So what happens is these dense stands provide an ideal microclimate underneath for the ticks, and it's also great host habitat. So you've got habitat and climate conditions, and so you can build up big tick populations there. And so studies done looking at reducing and long-term management of barberry here in Connecticut, we found that it significantly reduced the abundance of infected ticks. And in other studies, they actually found that removing the honeysuckle decreased deer activity in the numbers of Ehrlichia infected Lone Star ticks. So this invasive vegetation is something, aside from other effects, is also a tick risk factor. Spraying is probably still the principal method of trying to control ticks, particularly around residential properties. And this could include carbamates, pyrethroids, some biopesticides. We've done a lot of work looking at microbial, or what you call biochemical type alternatives to synthetic insecticides. The MET52 illustrated there is a fungus, and you can see a female tick killed by this fungus. And this chart here is basically a summary of research that's done over the years looking at the various different chemical treatments for tick control. You can see that the pyrethroids up at the top, bifenthrin and cyfluthrin and deltamethrin, tend to provide extremely high levels of control for many weeks. Carbaryl, or 7, either as sprayed in granules, also tends to be fairly effective for weeks to a few months. And then you have your 25B products. These are the EPA listed safe compounds. Anybody can formulate these materials into a product for deer repellent or tick control or whatever. They do not have to prove its efficacy, okay? So a couple of rosemary oil products, actually, which was IC2, was found to be actually fairly surprisingly effective. You can see 73, 95% to 100% control at the higher rate. Unfortunately, that product is no longer available. I did some sprays with garlic. I found average about 50% repellency for just a couple of weeks. Another product containing pyrethrum and thyme oil and so forth had high levels of knockdown, 99% with 72% residual control. But what a lot of companies are pushing right now is cedar oil products. So the limited testing that's been done so far, a couple of products that were evaluated at the University of Rhode Island, they only got 5 to 6% knockdown and very little residual activity. Another cedar product that was tested by a colleague of mine on Long Island, he got 31 to 40% control with it. Not that great. So just a caution that cedar oil is not the really high effective alternative, natural or organic alternative to traditional insecticides at this point in time. Another approach we've been looking at is host-targeted tick control focused on the mice and the deer. I'm not going to spend a whole lot of time on this, but there are bait boxes and tick tubes that target the mice. And then we've also done a lot of studies looking at deer treatment, deer reduction, and deer exclusion, and examined its impact on ticks. Reducing deer populations is effective, but you have to get them down below 10 deer per square mile to have an impact on the tick populations. Quite low. It's been done in a couple of locations, Monhegan Island off of Maine, where deer were introduced in the 1950s for hunting, were completely removed from the island, and now they have virtually no ticks, except for a few that come on migrating birds, for example. And I've done some other deer reduction studies that have actually shown an impact on tick populations as well. So what we're looking at a lot more now is integrated tick management, trying to combine some of these techniques, either host-targeted as well as different kinds of sprays and so forth. And I don't have time to go a lot into these, but there are 2 review articles, one of mine and one by a colleague's, that are in the Journal of Integrated Pest Management. and these are open access. So anyone here listening to me can go to that journal and find these articles if you want to read them on the tick control. And also as well, there's my tick management handbook, which is available on the Connecticut Agricultural Experiment Station website. A lot of the basics in there are still good. I published this in 2007. I'm working on an update revised version, but I'm not sure exactly when I'll get that out, but I am working on it. So just to give you an example of an integrated project, we did one in Reading, Connecticut. That's been published. We reduced the risk of encountering an infective tick by 66% by spraying that Met52 fungal biocontrol product, as well as using the fipronil bait boxes. So this is a study in Guilford, Connecticut, where we're looking at different combinations of a 4-poster. It is a feeding station for the deer, which you see here, and the paint rollers are treated with permethrin. So when the deer come up, they rub up against it, and the permethrin will kill any of the ticks that are on the deer. So the combination of the deer station, the rodent bait box, and spraying that Met52 in different combinations, we're looking at the impact of this integrated combination on reducing tick population and residential properties there in the neighborhoods in Guilford, Connecticut. We started that in 2017 with baseline sampling, and then we actually did the treatments in 2018, 2019. They'll be repeated in 2020. A lot of times I get questions about the Lyme disease vaccine. And so the original LymeRx vaccine was taken off the market in 2002 mainly because of unsubstantiated allegations that the vaccine was causing Lyme-like condition or symptoms. But in any event, there is a company, Valneva, is working on a new human Lyme disease vaccine. They've completed their phase 1 trials with it, so there was no safety concerns associated with that first stage. And they're in the process of 2 planned phase 2 studies in Europe and here in the United States. And they expect to have some of the early data from the phase 2 study here sometime in 2020. And then from there, they would go to the larger phase 3 trials. So we may get a human vaccine for Lyme disease again, but it's still a few years away. One of the things as I work here toward the end, I want to point out there is a federal tick-borne disease working group. The charter for this working group was approved by the Secretary of Health and Human Services back in 2017. And this working group was authorized by Congress for a total of 6 years. The charter defines how this group is structured, and it was provided by the 21st Century Cures Act, and it's renewed every 2 years. So the first report of this tick-borne disease working group to Congress was put out in 2018. This is available online. So if you're, and it's a really good summary of many aspects of Lyme disease and other tick-borne diseases. So just simply search online for Tick-Borne Disease Working Group 2018 Report to Congress. The group continued to work. There will be another updated report to Congress looking at aspects that were not necessarily covered by this first report. I was on the tick control subcommittee of that, and that report, of course, won't be available yet. But I suggest that you look it up. So that's a quick summary go through in the time that we allocated for this. I'll conclude here with this little prayer from somewhere around 1856, from red bugs and bed bugs, from sandflies and land flies, mosquitoes, gallinippers, and fleas, from hog ticks and dog ticks, from hen lice and men lice, we pray thee, good Lord, give us ease. Thank you very much, and I think we're going to somehow handle questions now, Brett.
Yeah, so thanks very much. We are getting pretty close to our time. It looks like in the chat, the one question, should I get chickens to eat the ticks, is one. And then somebody said, this was wonderful, thanks very much. And then another question was, when doing your check, should you be checking under armpits and in near crotch, things like that?
Okay, so I'll start with the tick check first. The black-legged tick has been recovered from anywhere on the body. And I will say, keep it simple. By when I say anywhere, I literally mean anywhere. So do need to do a thorough body search when you're looking for the ticks, okay? American dog ticks, about half of those do tend to be found attached around the head area and then elsewhere on the body. But the black-legged tick, you can find it attached anywhere. As far as the chickens go, there's very little support for that. There were a few little anecdotal studies that were done with guinea fowl, and you remember they're birds. The ticks will feed on those birds as well. So there's just not a lot of data on that.
Dog ticks versus deer ticks for doctors. I'm not sure exactly what that question means.
Repeat that, please.
ID dog ticks versus deer ticks for doctors.
ID, okay. So basically, all right. So American dog ticks. It's a common tick that we have here.